This is, as we said already last week, our first virtual block course. And so normally you would be in the lab um, culturing stem cells, differentiating neurons in real life. So what we're going to try and do today is introduce a 1.5-day module, so all day today and half of tomorrow, where we try and simulate some of the experiments that you're going to do, you would have done in the past, but you'll get the same output and then try and analyze the data. So if I could just ask people to turn on their videos, that would be great. It's nice to see people instead of just a blank screen. Thank you. So what I would like to do this morning is just give a very brief introduction to stem cells so everybody's on the same page. And some of the ideas that I'll introduce are going to be important for the rest of the experiments. So it's nothing in depth, it's just so that you understand the concepts. And then after that, at the end of the lecture, Luca is going to give a very brief introduction to Kahoot, which is an app some of you may be familiar with, which we'll be using in the rest of in the modules. So you can either use it online or as an app. And then you'll have another lecture, and then you'll break out into tutorial groups later on. So now I'm going to go to my slides here. Okay, oops, that's good. Let me just hang on one second. Sorry, I need to share my screen. That would help, right? Share screen. Oh, okay, there we go. Okay, so you should see my screen. And just get this on. Just getting all my windows together and then we'll start. One moment. Okay, so if you have a question, please raise your hand. I'll try and monitor that. If I'm not noticing, you can send me a quick chat, but I'll do my best to watch the participants as we're going along. Okay, so I'd like to tell you, just as I mentioned, a little bit about stem cells. So stem cells are really remarkable cells, and they're the reason we exist. So we start out as embryos and develop by these stem cells giving rise to more and more differentiated cells that then give rise to the full adult organism. So stem cells give us great insight into the biology of development and also of tissue homeostasis. But they're also of wide interest now for therapeutic reasons because there's great hope that they can be used to heal damaged tissue. And then another important aspect about stem cells is that they can also contribute to cancer if their tight regulation goes awry. And so the stem cells themselves can give rise to tumors. And within tumors, you have cells that can behave as stem cells. So together, these are really interesting reasons to understand more about stem cells. So today we're going to go over just very, very briefly the different types of stem cells, focusing on adult stem cells and pluripotent cells, just to understand where they come from and some of their key properties, such as their potency and their characteristic features. And then second, we're going to touch on how stem cells can be used moving into the future and why this is such an exciting area of research. So the first concept that's really important is that of potency. So what's potency? Potency, very briefly, is just the number of fates that a single cell can acquire. So potency decreases with age. So when you're during an embryo, the cells are more potent than in an adult. And the cells which escape this rule of decreasing potency are stem cells. So among the potency, there are different types of potency. We have totipotent cells. So these are cells that can give rise to all possible fates. So namely all the cell types of the body, including extraembryonic tissue. So things like the placenta. Pluripotent cells can give rise to many, but not all the fates. So they also can give rise to all the cell types in the body, but not the extraembryonic tissue. Then we have multipotent cells, which can give rise to many different cell types, but not all the cell types in the body. And then finally, we have bipotent or unipotent cells, which can give rise to either two or one type of cell. So the other feature of stem cells is they're classically defined by two key properties. The first is that a stem cell will divide to give rise to another stem cell. And so this property is called self-renewal. And this allows the stem cells to persist throughout life. The second key feature of stem cells is that they can undergo differentiation. So this means they become more mature and more specialized. So if we step back now and look at development, we can sort of put this concept of potency onto a timeline. So at the very earliest steps of embryogenesis, the sperm fertilizes the egg and gives rise to a zygote. This zygote will then divide and grow to give rise to something called the pre-implantation blastocyst. And this is essentially the inner cell mass inside this blastocyst. These cells give rise to the embryo. So these cells will divide and undergo gastrulation, which gives rise to the different germ layers, such as the ectoderm, which will give rise to skin and to the brain, mesoderm, that gives rise to muscle, and the endoderm, which gives rise to many internal organs. So after gastrulation, stem cells will continue to divide and give rise to the fetus, and then the fetus will eventually give rise to an adult. So these earliest cells are pluripotent and totipotent, and then you have somatic cells in the adult. So these are fully differentiated cells that no longer can act as stem cells under normal conditions. So now, if we map the types of stem cells as through development and adulthood, what we can see is that these earliest steps of development, so the zygote stage, are totipotent. So stem cells from these stages can give rise to the embryo and the trophectoderm or placenta. Then, as you progress along, you come to the pre-implantation blastocyst. The stem cells found in the inner cell mass here are pluripotent. So they can give rise to the whole embryo. And in vivo, they're a very transient population of cells. Then, as you progress along development, the stem cells become more restricted. So that you often find multipotent stem cells that exist in different organs, and they're usually tissue specific. So during embryonic development, because the embryo or the fetus is growing so quickly, these stem cells divide very rapidly. But in the adult, many of these are quiescent, so they're dormant, and they give rise to an actively dividing cell and then re enter quiescence. So this is a big difference between development and adulthood. So let's talk briefly about adult stem cells then. So adult stem cells are found in many different organs of the body. Among them were the first identified stem cells in the blood system. We have stem cells in the skin and the muscle, the intestine, and surprisingly also in the brain, which was thought to be a very static organ. So why do we need these stem cells? It's because differentiated cells are constantly turning over. So, for example, in the intestine, the mature cells last only a few days, so they have to be continuously replaced. Blood cells have a lifetime of only about 3 to 4 months. They also need to be replaced. And skin cells can live 2 or 3 weeks. So, stem cells then in these different tissues are there to maintain the homeostasis of these organs, so give rise to new cells that replenish them. They can also be recruited for repair of a tissue after injury. And many of these cells are multipotent. And an important idea is they live in complex niches or microenvironments that support their lifelong maintenance. So, how do adult stem cells contribute to tissue throughout life? And this is true for most adult stem cells. You have a rare population of stem cells found in the organ, and these cells can undergo self renewal, and they typically divide slowly, and they have a very high potency. These stem cells then give rise to so called progenitors or transit amplifying cells that divide very rapidly, but they're more committed than a stem cell. And under normal conditions, they don't typically self-renew. They divide a few times and then disappear. These transit amplifying cells or progenitors then give rise to progressively more restricted cells that become fully differentiated and they no longer divide. So these are post-mitotic cells. So these adult stem cells then, as I mentioned, live in complex niches or microenvironments, and these provide support and signals that regulate self-renewal and differentiation. So here in blue, you have a stem cell, and in orange, you have niche cells. And there's actually a lot of signaling that occurs between the stem cells and their niche. But the two key types of information and regulation that are provided are via direct contact. So in this case, the stem cells directly contact niche cells, or for example, a basement membrane that's made up of complex extracellular matrix, and this can bind growth factors and present growth factors to the stem cells. The other kind of factors are when niche cells secrete soluble factors or diffusible signals, and these can then influence the stem cells remotely. So, one of the key open questions in many fields is which niche factors influence stem cells, and can we recapitulate these in vitro to try and model this in vitro? So, finally, stem cells can undergo different types of division. So people often think of stem cells as undergoing asymmetric divisions, where a stem cell gives rise to another stem cell in a differentiated cell. And this means that the stem cell number stays the same over time. However, under certain conditions, stem cells can undergo symmetric division, and this leads to an increase in stem cell pool. And this often happens during an injury state. And similarly, some stem cells can undergo division to give rise to two differentiating daughter cells. So in this case, the long-term consequence would be a decrease in the stem cell pool. So in the adult, as I mentioned, you often have quiescent or dormant stem cells and actively dividing stem cells found in the same tissue. So why is this? So typically, a quiescent stem cell will become activated, give rise to an actively dividing stem cell that self-renewed, and then these give rise to committed progenitors that then give rise to differentiated cells. And one interesting theme that's emerging, however, is that even within one organ, there are many different types of stem cells. So there's a great diversity or heterogeneity amongst the stem cells that reside there. And the meaning of this heterogeneity is an area of active research. Can they make different cell types? Why do we have these different pools of cells? So after injury, what happens? Well, typically, dormant stem cells become activated to give rise and regenerate the tissue. But it's also been seen that stem cells can exhibit plasticity. So one cell type might turn into another, or even committed progenitors may revert back to a stem cell state to completely regenerate the tissue. So all of these questions are really interesting questions that are actively being pursued in the field. And tomorrow morning, I'm going to give a very brief research overview of some of the questions that we're trying to understand in the brain about adult neural stem cells, which really represent an unexpected form of brain plasticity. And so we're curious as to what these cells do, what regulates them, and why do they exist. So now I just want to cover briefly pluripotent stem cells that are going to be the cells that you'll be working with in your virtual embryonic stem cell module. And there are two types of pluripotent stem cells that we're going to talk about. The first are so-called embryonic stem cells, and the second are induced pluripotent stem cells. So to understand where embryonic stem cells come from, we have to go back to development. So here we have an egg that's been fertilized. It then proceeds along the oviduct and begins to divide to a 2-cell stage, 4-cell, 8-cell stage. Then it becomes a morula and it moves into the uterus. At this point, until the morula stage, All of these cells are totipotent, so they can all give rise to every single cell type. Next, you have compaction, and here we have the inner cell mass in the preimplantation blastocyst, so this right here. And it's at this stage that the embryonic stem cells can be isolated. So here we see in a schema form the inner cell mass from the blastocyst. These cells can be harvested and cultured then in vitro and propagated forever as embryonic stem cells. So these cells are fully pluripotent. They really exist only in vitro, and they can self renew indefinitely if they're given the right conditions. So, how is it that these cells can be maintained as a pluripotent stem cells? Well, they express a cluster of different transcription factors, which are known as the pluripotency transcription factors. And some of the most famous are OCT4 and SOX2 and NANOG and ESRRB. And all of these are co expressed uniquely in these early epiblast cells that give rise to the ES cells and embryonic stem cells. And they're highly interconnected in a really robust network so that they reinforce the expression and therefore can maintain this pluripotent state. So, pluripotent stem cells can give rise to cell types of all three germ layers. So, they can differentiate into endoderm, into ectoderm, and to mesoderm in vitro. And in vivo, they're able to give rise to all the different cell types. So, one way that people test this is to see whether embryonic stem cells, when they're transplanted into a blastocyst, can contribute to different cell types in a mouse. So, here we have an albino mouse that was given embryonic stem cells from a brown mouse, and you can see the brown cells contribute to different tissue, and in fact contributed to the germline because here are pups from this mouse and they're completely brown. So it means that they gave rise to all the different cell types in the body. A second assay that's used is teratoma formation, where you transplant embryonic stem cells and then you form a little, almost a tumor, where you have all the different cell types within this teratoma. So, embryonic stem cells then are really powerful cells. They can give rise to all the different cell types in the body. But then the challenge is, how do you direct them to differentiate into the cell type of interest? And so, what's emerged is that we can do this if we follow the sequential developmental path that cells normally undergo. So, we can imagine here we have embryonic stem cells. If we want to make a neuron, first, we have to differentiate the cells into ectoderm and then into neuroectoderm, and then follow down this sequentially more and more restricted paths until you reach a mature neuron. Likewise, you can make them go down a different path and make cardiomyocytes. So I just want to take a moment and show an example of how we would go from embryonic stem cell to form motor neurons. So motor neurons are found in the spinal cord. This is a developing mouse embryo, and this is the front and this is the back. So spinal cord cells here, and you can see that the progenitors or stem cells in this area would be exposed to sonic hedgehog signaling from the ventral side and retinoic acid signaling from the dorsal side. And the co-expression of these two signaling factors induces these motor neurons over time. So in a dish, how would you do this? You would take your embryonic stem cells, then you aggregate them and form embryoid bodies. And the beauty of embryonic stem cells is you can make many, many of these. Then you expose the embryoid bodies to developmental patterning signals like sonic hedgehog and retinoic acid. And then you can induce them towards a neural fate. And then over time, if you let them mature, they turn into beautiful branched motor neurons in a dish. But are these cells really like motor neurons in vivo? So one could test this, for example, by isolating these cells and transplanting them, in this case, into a developing chick embryo. And you can see here in green, the embryonic stem cell derived neurons exactly follow the trajectories that the normal motor neurons do in red. So these cells are fully functional, and you can actually record using electrophysiology and show that they really are integrated. So embryonic stem cells then allow us, are pluripotent cells that can give rise to all different cells in the body. And they're considered still the gold standard. They don't have genetic abnormalities. They can be expanded ad infinitum and differentiated. However, there are some ethical issues, especially when one thinks about humans, because they have to be derived in vitro. These embryonic stem cells have been used for many years to do targeted genetic modification and provided many, many mouse models to understand disease modeling. However, more recently, a new type of stem cell has emerged that's allowed us to actually test human cells more directly. And these are induced pluripotent stem cells. So, ES cells, embryonic stem cells, were thought for a long time to be the only type of pluripotent stem cells. And this idea was that differentiation is irreversible. As you go from a stem cell to a progenitor to a differentiated cell, the potency of these cells decreases, and it was thought they could not go back to a pluripotent state. But now we know that actually you can take a fully differentiated somatic cell and reprogram it to a pluripotent state. And this discovery actually was awarded the Nobel Prize in 2012 to two remarkable scientists, John Gurdon and Shinya Yamanaka. So, John Gurdon had done very early experiments in the 1950s looking at frog cells. And what we're going to talk briefly about today are the more recent experiments by Shinya Yamanaka, where he was able to do this with mouse and human cells. And they got this prize for the discovery that mature cells can be reprogrammed to become pluripotent. So the idea is that you can take a skin cell or a fibroblast or many other types of differentiated cells and reprogram them by delivering the so-called Yamanaka factors to them. And in this case, these were OCT4, SOX2, KLF4, and c-MYC. So you'll recognize that several of these are actually these pluripotency factors. So you can reprogram the cells. They go to a fully pluripotent state, and you can then direct the differentiation of these pluripotent cells into whichever cell type you like, such as a differentiated neuron. And the advantage of these is that these cells are patient specific, so they won't be rejected if they're going to be used for transplantation. Excuse me. They can be used for disease modeling and for screening drugs directly on human cells. So if we put this together, a new model is emerging for how stem cells can be used. We can isolate a skin cell from a patient. You can reprogram these skin cells into induced pluripotent stem cells and then direct them along different lineages to form different types of differentiated cells. And these cells can either be transplanted back, perhaps, or they can be used for disease modeling to understand why a disease occurs, to screen for drugs, and to do toxicity tests, which are often very difficult to do without human cells. Another major advance that's recently occurred is moving from two-dimensional to three-dimensional cultures. So the problem with two-dimensional cultures, which were used for a long time, is you miss some of these more 3D cell-cell interactions. And now people have identified or developed methods to make so-called organoids, which are really self-organizing three-dimensional structures that come from stem cells. And you can make these in the same way by simply exposing pluripotent stem cells to different developmental signals, and then you can get organoids of almost every type. Here we have an intestinal organoid, a kidney organoid, a cerebral or brain organoid, and optic cup organoids. What's amazing about these is you can begin to understand very early human development and also model disease. This is just to show that many different organoids have been made from different organs in the body. And here are some examples. And you can see that they don't completely resemble the mature organ, but they have many features. So here we have a mini brain organoid. Here we have a stomach organoid, the typical folding. And here we have a skin organoid with developing hair, which one can actually transplant, and you can see give rise to hair. So, in the last couple of minutes, I just want to touch on how some of these stem cells can be used first to cure disease cells, and second, if we can repair mutations that happen, just to illustrate the power of stem cells for regenerative medicine. So, I'm going to just briefly touch on two examples. The first is a really nice example of what we consider personalized medicine. So, really for the individual. And this case talks about a disease called cystic fibrosis, which is due to a mutation in a channel. And these patients have problems with their lungs and their digestive system because the channel won't open. And this leads to a mucus buildup and bacterial infection. So you can actually isolate a biopsy from a patient, and in this case, you could grow intestinal organoids and screen drugs. And they were able to actually look for drugs that allowed the channel to open and allow the proper flow of ions. And this drug was then given to the patient and was able to cure them very rapidly. So, this is a great example of how you can screen for drugs that are specifically targeting one mutation and then can then be used for a patient. The second example is, can we actually repair a defective gene in a cell? So here is a terrible disease called epidermolysis bullosa, and this is caused by mutations in laminin genes. And these are required for anchoring the epidermis, or the outer layer of skin, to the tissue below. And if you have a mutation in these laminin genes, the skin blisters and just tears off at the slightest touch. And the only treatment is daily bandaging of the wounds. So it's a devastating disease. And in a remarkable study, they were able to take unblistered skin from a patient and make stem cell cultures. And they were able to use viral vectors to correct the mutation in the laminin gene and then grow epidermal stem cells and have them form these large transgenic sheets of skin in vitro, and then transplant these huge sheets of skin onto a patient. And over time, and this is now several years since this has happened, the patient was completely cured. So they all of the stem cells are there. They continue to make the healthy gene, and the skin is repaired. So this is also another beautiful example of how gene therapy, in some sense, can be administered together with stem cells to really help patients. So there's a lot of hope then for using stem cells to model diseases, for drug screening, to replace cells, and perhaps even to harness endogenous stem cells. So what are you going to do? So in the virtual block course then, you're going to turn embryonic stem cells into neurons. And you're going to do this by culturing in virtual world and direct neuronal programming of embryonic stem cells. That's going to happen today. And then tomorrow in the morning, you're going to characterize these embryonic stem cells and their progeny based on their morphology, on their RNA expression, and on their protein expression. So just very briefly, how are you going to do this? I told you before a lot about how typical embryonic stem cells can be programmed or directed to undergo differentiation to a neuron. What we're going to do in this block course is actually bypass all of these developmental steps, going straight from a stem cell to a neuron by overexpressing a transcription factor. And this is an example of so-called direct programming. And we're going to use a cell line that overexpresses a transcription factor important for neuronal differentiation called neurogenin 2. So this is going to be the model you're using. You'll explore this with your tutors this afternoon, going from an embryonic stem cell into a mature neuron. So I'd just like to introduce the tutors. We have Zaina, Valerie, Anna, Fabrizio, Violetta, Thomas, and Luca, who will be working with you on the ES cell in different parts of it. And then Karina is going to be working on the brain anatomy on Wednesday. So just to remind you then, this is the schedule we have for today. We have my lecture. After this, Luca is going to give a very quick introduction to Kahoot. Then we'll have a break until 10. Then Violetta is going to give an introduction lecture to cell culture so that you understand how to work with cells and the culture. And then we're going to have a break. And then you're going to separate into 6 different groups and work with your individual tutors this morning. And then in this afternoon to touch on 2 different modules. And then tomorrow morning, you'll have a brief research lecture by me. And then you'll start with the molecular characterization of the stem cells and progeny, working in 3 groups. So, does anybody have any questions? Stop my share. It's all clear. You can raise your hand if you have a question or wave. No? Okay. Super. So then I ask you to hang on for 1 second. And then Luca, Luca, are you there?
Yeah, I'm here.
I just realized, can you try sharing your screen? If not, I may have to change some settings. It works. Perfect. Okay. Yeah.
All right. Good morning, everyone. My name is Luca. I'm going to give you a very brief introduction to Kahoot. This is a software or this program where you can do these real-time quizzes with your phone. Some of you may already know it. So basically, can you see the correct thing? Yeah, perfect. All right. So what is Kahoot? It's basically this program where you can have these short quizzes which you can answer via your phone. And so this is going to be more an interactive part of the block course. And so what I'd like all of you to do is to take out your smartphones.
Luca?
And yeah.
We see your, we do not see the presentation view.
I was seeing presenter. Yeah, that's better. Yeah.
Great. All right, perfect. Thank you. So if everyone could go take their smartphones and type in this www.kahoot.it. So if you could do so now, or those with the app, if you could open the app. So if everyone could give me a reaction or a thumbs up or something so I can see. Right, perfect. So don't type in this game pin. This is just an image of another one. So just go to the start screen at the moment, and then I'll just kind of lead you through it, how it works, and then we can test. We can do a test one very briefly. All right. So when you go to this screen, you have the opportunity to see this pin. And let's see if this works now. And then in the end, when you type in this pin, you're asked if you can write your own name or any kind of name that you want to, or you'll appear here.
But I tried it and it's written that this pin isn't recognized.
Exactly. So this is just an image. This is not a real game. This is a PowerPoint of a previous game that I made. So don't type in this pin yet. So I'm just going to lead you through the Kahoot itself, and then we can test it. All right. So as soon as you type in your name, then the host, which is in this case me, is going to share my screen and start the game. So what it looks like is what you'll see on your Zoom screen is the question. So what is 1 + 1? And then after a few seconds, you'll be led to this screen. So on the top, you have the question again. Then on the side, you have the time remaining. So in general, it's between 20 to 30 seconds per question. And in the bottom, you have your 4 answers. So 3, 2, or 1 and 5. So this is on your laptop screen or your computer screen. On your phone, it looks like this. So you just have a red, blue, yellow, or green, or triangle, and so on icon that you then press to give the correct answer. So in this case, what is 1 + 1? On your phone, you would press the blue rhomboid. Okay? And then once everyone has put their answers in, the correct answer is going to come up. I mean, it's really basic and simple. So let's just test this briefly. Switch to here. All right. So now if you could all enter this pin. So it's 182665. Exactly, and then enter your name. Great, perfect. I think that's very good. All right, so is everyone inside? Is anyone still missing? No? All right, we'll start the game. I think you all know the game quite well. Great. Okay. I mean, that's basically it. I mean, you could all calculate 1 + 1. Was there anyone that had difficulties or there was a problem? I think everyone got it more or less. All right. So one more question, and then that's already it. I mean, this is just to show you the software. So Simona, congratulations. You were the fastest person to click on the correct answer. Few more seconds. Okay, perfect. So I think we all got how it works. And now for the podium. Nicole, congratulations, you're 3rd place. Lucas, you're 2nd. And the winner is Atik. Congratulations. All right. I think that's all for my part. And I wish you good luck and have fun for the rest of the day as well.
Oops. So just, we're going to take a little break now. You're going to be using Kahoot in some of the modules, so it'd be great to just keep your smartphone handy. And so we have a break now until 10 o'clock, and we'll just have a brief introduction to cell culture then, and enjoy your time in the next day and a half. Thanks a lot.